Hello students, welcome back. Today I am Radha from DPS Alwani Lamachor going to start another topic of the same chapter which we were discussing in the last class, that was the biotechnology. Okay, so in this chapter we have done the introductory part. So today we will start that how we will go for the genetic engineering. Okay, we have done two principles, that was the genetic engineering or the bioprocess engineering. Okay, so if I will talk about the genetic engineering, it means that we are manipulating at the gene level. Okay, for that what should be the tools which will go for it? So already we have discussed about the one. That one was the restriction enzymes, cloning vectors, competent host organisms. So what does it mean? That if I have to transfer any gene of interest, if I say that it is my gene of interest, okay, which I acquired, and I have to incorporate in any host organism, okay, in any host organism. So for what is required, I want that it should be moved from here to here. So who will take this gene of interest into this host organism? So what is required? We required the mediator part. What is it? The vector part or the vehicle which will take this gene of interest here, and after that it will send into the host organisms, right? So we have to manipulate in this structure. So how I will manipulate? With the help of the genetic engineering. And the first thing which is required here, the restriction enzymes. Okay, so this gene of interest is coming from where? From the whole DNA. Okay, for example, if it is a whole DNA, okay, if it is a whole DNA and I want the specific part of this DNA, so what is required? It is required that I have to cut this DNA. Okay, to cut this DNA, what can be the tools? Okay, so the tool is what? The restriction enzymes, and these restriction enzymes we are going to discuss today. Okay, so now if I will talk about this restriction enzyme, so what are these? The another name is the molecular scissors. Okay, scissor mean to cut something. When I will talk about cutting at the molecule level, okay, when I will cut the DNA molecules, what will happen? We will use these restriction enzymes. That's why it is known as molecular scissor. So with the help of these molecular scissors, we can cut this DNA, okay, and we can isolate our gene of interest from the whole DNA. Okay, likewise, if I have to incorporate here in this DNA, in which DNA? The DNA of the vector 1, because this vector will take here into the host. Okay, so what is required? I have to cut the DNA, the gene of interest from here, and then I will uh, provide it into the vector 1. So what I have to do? I have to cut this part also. Okay, so what is required? Then again molecular scissors are required, whether we are cutting the DNA from the gene of interest, okay, or we are cutting the DNA from the vector. We need the molecular scissors, and these are what? The restriction enzyme. As we will cut it here, okay, if I am showing with the another one, this is my gene of interest which I have isolated from the whole DNA. So what will happen? This will move here and it will join with the DNA of the vector 1. Okay, so likewise what will happen? We can move this whole DNA into the host organisms, and after that it will replicate here and we will get our whole gene which is of our need, right? So in this genetic engineering, we are going to do what? We are going to cut, we are going to stick, and then we will get the copy, right? So the first thing, as I have said, restriction enzyme, some points we can discuss it here. That as I have said the first time, okay, these are the molecular scissors. So how we have isolated at the first time? So the two enzymes from the E. coli. E. coli is what? Bacteria. So first of all, when they have isolated in the 1963, what they have found, that this E. coli, okay, this E. coli, I'm rubbing it here. So this E. coli is a bacteria. It contains two types of enzymes. Okay, what are these two types of enzyme? The first enzyme is able to add the methyl group to the DNA. Okay, the first thing what it will do, the enzyme which is present inside this E. coli, it is responsible to add the methyl group to the DNA, as well as the second one is of our use. Okay, second enzyme which is present in this E. coli, it is responsible to cut the DNA. Okay, so this DNA is having the enzyme which is present in this E. coli, it is able to cut the DNA into different segments. If it is having the ability to cut into different segments, what we will call it? We will call it the restriction endonuclease. Okay, what is it? Restriction endonuclease is what? To cut the DNA, and which is having the tendency to cut it? The E. coli, because E. coli contain some these types of enzymes. Right here. Now the question arises that this E. coli, okay, what it is doing, if any of the bacteriophage is what, which is having the ability to attack the bacteria. So we know different viruses. Okay, viruses, what they do, they remains their body outside, and the DNA which is present in the inside the bacteriophage, it gets inside the bacterial cell. Okay, and due to that, why? Because it is having the ability to cut the DNA. What it will do? It will restrict the growth of this bacteriophage inside this E. coli. So it is having this ability. So what they have done? They have isolated this enzyme from the E. coli. Okay. So likewise, earlier the first time it was isolated in 1963, and the first restriction endonuclease which they have isolated, it was the Hind2. Okay, make sure that you must memorize that Hind2 is the first restriction endonuclease enzyme which they have isolated. Okay, and the scientist name is Wilcox and Kelly in 1968. So the first time they have isolated, or they have made the Hind2 restriction endonuclease enzyme. Right here. Now, same thing, if I will talk about this Hind2, it is having the specific ability. Okay, if I will talk about any of the restriction endonuclease enzyme, it is the characteristics of this enzyme that it always cut at a specific site. If it is a base pair of 6 base pair sequence, okay, if I will talk about any of the DNA, and from that part I will take the 6 base pair, so these restriction endonuclease enzyme will cut at the specific point, and each restriction endonuclease having the different recognition site. Right? So what I am saying here, that Hind2, which is also a restriction enzymes, what it does, it always cut the DNA at a particular point. Okay, at the particular point of the 6 base pair sequence. For example, if I say that this is the base pair sequence, okay, so what happens, it is having the specific point, it will not cut from anywhere. For example, I'm writing from here GAATTC. Okay, so this is the first strand, and the second strand is what, then CTT, then AAG. Right, like this. So always and always this Hind2 will cut between the bond of G and A. Okay, it does not cut the bond between any other sequences. It is having the ability to cut the bond between G and A. So it will be what, it is very specific, and these specific sites are known as what, recognition sequence. Okay, so it always recognize at a specific sites. Right, so we can say about the restriction enzymes, these are having a specific recognition site at which they will cut the DNA strand. Right, this is the point. And likewise, what happens, that we have isolated different enzymes. We have isolated different restriction endonucleases from different bacterial cells. So each of the restriction endonuclease follows the same thing. Okay, so for your syllabus, it is what it is that we have to understand this Hind2, because it always cuts the bond between G and A. Right here. So now what happens that if I will talk about this Hind2 having some different recognition site. Okay, and likewise we are having one more restriction endonuclease, that is eco R1. Okay, what is it? Eco R1. Right now, which I have told you that this bond between G and A will be break down by which type of restriction endonuclease? Eco R1. Okay, and if I will talk about the general characteristics of the restriction enzyme, that it is having the specific site at which it will cut the DNA sequence. Okay, so here we have done what is the recognition sequence, and it is followed by the restriction endonuclease enzymes, right? So here I have shown the mechanism how all these things happens here. So for that we are having the vector DNA. Vector DNA is what? At which the DNA or the gene of interest will move here. Okay, if I have any particular gene, okay, if this is a foreign DNA for me, okay, and I have to take a specific part from this foreign DNA into this vector DNA. So what I will do, I will use this restriction enzymes, right? So here we have taken the example Ecor R1. Okay, we have not taken example here Hind2, but Hind2 was the first one. That's why you have to memorize this one, right? So here we have talked about the example of Ecor R1. Whenever we use this Ecor R1, as I have said, it always breaks the bond between G and A. Okay, anywhere. So what we have done, we have taken the vector DNA. Okay, what I have to do, I have to cut this part. Okay, how? And we have to take this gene of interest from the foreign DNA, and this gene of interest should be incorporated in this vector DNA. Okay, in the previous class I have shown you that if this plasmid, okay, plasmid can be also the vector. So what happens, I have to cut this plasmid. Okay, I have to cut this plasmid, and for example if it is my DNA, and from this DNA I have to take only this part. So what I have to do, I have to cut that much part and I have to add this part into this plasmid, right? So same thing, this vector DNA can be the plasmid, and the foreign DNA is that DNA which is of our use, and I have to incorporate into a particular vector DNA, right? So for that, what we have done, this is a sequence. Okay, the thing which I have shown here with the plasmid, these are the two strands, right? So if I will show these two strands like this, what will happen? This can be like this, okay, and here one end can be 5 prime and 3 prime end, another one can be 3 prime as well as 5 prime end. Okay, now if you will see the sequences here, again 6 base pair sequences we are having here in the vector DNA, as well as 6 base pair sequences in the foreign DNA as well. Okay, now same sequences, if we are using the same endonuclease enzyme, what it will do, it will cut at the specific recognition site in both of the DNA, right? So we have used here Ecor R1. What it will do, it will break the bond between G and A here, from here as well as from here also, right? So in the same way, what it has done, it has break down the bond between G and A here, okay, as well as this part. Okay, so what you can see here, it will be somehow the stretching hand. How? Because if I will cut this part here, so this G will remain at a free arm. Okay, here also it is having the free arm. So these stretching sides you can see at the cutting end of the DNA. Okay, so the first thing which we have done, we have cut the DNA strand with the help of Ecor R1. Now if you will cut it, what it will look like? As I have said, it will break down. What will happen? that the whole sequence will get lost from this side. Okay, so what it will form? GCTTAA, right? That much is left from the vector DNA. If I will cut the whole part, okay, if I will cut the whole part, this G bond, so what you are having? This AATTC, okay, and again if I have broken down this bond, so what you are having here? This much. Okay, so what you have find out that these are having the different ends, and these different ends having the different base pair. Okay, on the basis of that we can say that these are the sticky ends. What you will call it? Sticky ends. Two types of ends are there, that is the blunt end and another one is the sticky end. What is the difference between these two here? That if I will talk about this blunt end, okay, in this blunt end, what happens that if it is GC, okay, GC, then GGCC, okay, here also GGCC, okay, like this. If we are having the chain, for example, what we are having, we are cutting between these two, okay, G with C, G with C, G with C, and here also G with C, then G with C. So what you are seeing here, we are taking the 6 base pair sequence G, C, or we can say change the sequence above C and downward G. So what is happening here? If you will cut this type of thing, so what you will get here, that same sequence this part also and same sequence this part also. So what it will form? It will form the blunt end. Okay, if you will go the strand of these two here, if you will go the strand of two here, what you will get it here? The blunt end. But what we require the sticky ends, because sticky ends can be stick very nicely so that we can get the proper end, right? So here the thing which we have prepared here, it is a sticky end because it is not having the same part. You can see here, if it will be the same part, how it will pair up with the DNA of foreign one. Okay, so it is required that there must be a complementary base pair sequence, and the bond which is present between these one, this is the hydrogen bond. Okay, and the bond between G and A is a phosphodiester bond. So we will break the phosphodiester bond, and with the help of hydrogen bond we will attach the two strands of DNA template right here. So what you can see here, we are having this GCTTA. So what is required for this TA? For this T, what is required? A is required. For this T, A is required, and for this A, T is required, and for this A, T is required. So from where it will come? It will come from this part right here. So if you will take whole of this part here, what you will get here? A, T, T, and this C, G. Now it's completed. So what we have done, we have joined these two and we got the recombinant DNA, and this is the ultimate aim what we have to do. We have to join our gene of interest into the vector DNA, then we can transfer it into the host organisms, right? So here we have completed how we can use this polycular scissors to cut it, right? Then some more important about this restriction enzyme is that how we have given the name to these restriction enzymes. Okay, so how we have given the name, you can say we are having different restriction enzyme. For example, EcoR1 which we have used here. So this EcoR1, how we have named it here. So this EcoR1 we have isolated from which bacteria? From the Escherichia coli. E. coli, Escherichia coli, or we can say E. coli. Okay, and this E. coli having a different strain also. So how we have given the, if I will talk about the first letter of the restriction enzyme, it is the genus name, that is Escherichia. Okay, and the two letters, the next two letters which is present in this restriction endonuclease, it is coming from where? From the species. Okay, first letter from the genus, then another two letters from the species, and this can be the strain, right? And the first, this Roman numeral shows that what is the sequence of the isolation. Okay, the restriction endonuclease which comes first one, it will get the first name in the Roman numeral, and which will come later on, it will get the another name or another sequence in a number. So we are having the EcoRAR1 restriction endonuclease, then EcoRAR2, same thing, Escherichia coli. Okay, R is that strain, and it is isolated at the next level after this EcoRAR. That's why we have given the name second. Okay, so these are the methods, and these questions may be asked in the examination, that what are the different ways by which we can name to these restriction enzymes. Okay, another important thing about this restriction enzymes is that these comes under which class? The class nucleases. Okay, and these nucleases can be of two types. What are they? Exonucleases and the endonucleases. Leases or but whenever we are talking about the breakdown. Okay, breakdown of what? Nucleotides. That's why we have given nucleases. So this exonucleases is what? Exo means outward. If it is a strand of DNA 5 prime and 3 prime and 3 prime and 5 prime, if there will be breaking down of bond from the extreme side, or we can say either from the 5 prime end or from the 3 prime end. Okay, if we are going to break the bond from the 5 prime end or from the 3 prime end or from this 3 prime to 5 prime, what you will call? Exonucleus. Endo means what? Inside. Okay, if these are the stand and we are going to break down the bond in from the between. Okay, we are not going to break down the bond from the end point. So what you will call it? Endonuclease enzyme. And this endonuclease enzyme we are using here in this mechanism. Okay, and this is another feature of this endonuclease, is that it always it is having the recognition site. Okay, it will break down at the specific site, and the specific site is present where? In this palindromic nucleotide. Now, what is this palindromic nucleotide, or what is this palindrome? Okay, so always and always if I say it has to break down, so this sequence should be palindrome. Okay, so what is the meaning of palindrome? The sequence of the base pair. For example, this is the strand. Okay, if this is the strand, so the sequence of base pair in both these two strand, it should be reading as the same. For example, if I will go from this 5 prime to 3 prime end, okay, and from this 3 prime to 5 prime end, what will happen? I will get the same sequence, right here. So same thing, if I will say that the sequence which you are seeing here, what is it? G, A, T, T and C, right? Then another sequence which you are getting here, C, T, T, then A, then G. Okay, so this is the sequence. So 1, 2, 3, 4, 5, 6. 6 base pair sequences are there. Whether if you will move from this side, if I will move from 5 prime to 3 prime end here, G, A, T, T, C. Okay, now if I will move from this side and I will read these sequences from here also, it will be the same. G, A, T, C. Okay, there will be no change. So these types of sequences are known as palindromic sequence. And in your NCERT, one example is given, Malayalam. Okay, if you will read from this side, again you will get the same one. If you will read from this side, again you will read the same. So these are the palindromic sequences, and on these palindromic sequences, this endonuclease enzymes act upon. Okay, it will act upon only and only in this palindromic sequence, and in this palindromic sequence they are having the specific site, and these specific site will be different for the different type of restriction enzymes, right? And restriction endonucleases enzymes. So here we have done that how they will recognize their site and how it will break down it. Okay, so that much is there in the restriction endonucleases. Now what we will do, whatever the things or whatever the genes or the sequences which we have cut it down here, it should be join out Okay, so what we have used here, we have used the restriction enzymes EcoRR1, and to stick these two things, what is required? DNA ligase. Ligase is what? Ligase is the sticking part which will stick these sticky end together. Okay, so what is required? We require the molecular scissors to cut it and the DNA ligase to stick it, right? Now, when we will get these cutting fragments of DNA, what is required? We require some more and more. Okay, we need to amplify the gene fragments. So we can use some technique here, and that electrophoresis is one of the method. So now here we will understand that what is this gel electrophoresis, by which we can amplify the copy or the DNA fragments, and we can remove out the DNA fragments from here. Okay, so the method is what? That is the gel electrophoresis. As the name itself indicates, here what we are going to do, we are using some gel, and that gel which we are going to use here, that is the agarose gel. Okay, so this agarose gel having some pores, through this what will happen, there will be some sieving effect, and due to that there will be movement of ions from one part to the another part. So we will understand that how all this work here, right? So the first thing about that, what happens if we have to separate the DNA fragments. Okay, what is the DNA fragment which we have cut it down with the help of this restriction endonuclease. So these fragments should increase the number, as well as these fragments should be needed according to their size. So how we can use this technique and why we are using this technique? We are separating our DNA fragments from their part based on their size. Very important thing. So on the basis of their size, we can isolate the different type of DNA fragments. Okay, it can be the larger one, it can be the smaller one. So here the DNA fragments can be visualized. So it is the next step, how we can see these DNA fragment, because these are the colorless. So we require some technique here. So before that, in this technique, what we are going to do, we are going to prepare a agarose gel bed. Okay, you can see here, this is the bed, what? Agarose gel bed. So in this bed, we are having different wells. Wells is what? In which we are going to fill the DNA fragments. Okay, so here having different fragments, 1 2 3 4. So different wells are present here, and what we are going to do, we are going to incorporate our DNA into these wells here. Okay, now the nature of the DNA is what? The nature of DNA always is a negatively charged. Okay, we all know about it, that DNA is a negatively charged. So if we have to move this DNA fragment from here to here, what is required? We need that we have to create some charge so that it can move easily. Okay, so how we can create this charge? With the help of the electric field. So we will apply the electric field here, so that this negative should move towards the positive. So we will provide some anode part to the opposite end, and this DNA negative charge will act as a cathode, and to the opposite of that we are having the anode part. Okay, as we will attach the anode part here, what will happen? These negative charge will try to travel from this cathode to anode. Is it fine? From this negative to the positive charge, and due to the sieving effect, as these agarose gel having some pores, so it will help them to travel in a proper way. So it will move according to their size. What does it mean? That if the size of the fragments will be larger, so they will not be able to move up to anode. Okay, and the size which are smaller, okay, the DNA fragments which are smaller, they can travel very fast. So you can see here the smaller size is present at the anode part, and here you can see that the size which is having more one, it is here only. So here on the basis of their size, we can isolate these DNA fragments easily, right? And we can use in our genetic engineering. So another thing what we are going to do, as we have provided the electric charge, we got the DNA fragments which are of smaller size. Okay, so on the basis of size we have categorized them. Now we have to, if we have to visualize these things, whether it is the larger size or the smaller size, how we will come to know? For that we have to go for the staining. Okay, because DNA is a colorless one, so we cannot see all these things with our eyes. So what is required? It is required that it should be stained with the help of ethidium bromide. So this question is important. Which of the compound we are using here to visualize the DNA fragments in the gel electrophoresis? That is the ethidium bromide. Okay, as we will stain with the ethidium bromide, then under the exposure of what? Ultraviolet light or the ultraviolet rays, it will be visible. Okay, so what is the condition? We have to stain with the ethidium bromide, and then we have to provide the exposure with the UV. Right? As you will see here, what you will observe, that these band will show as a color of orange. Okay, so orange color bands we can see here, which shows that these are the larger particles and these are the smaller one, or the larger fragments or the smaller fragments. Now what we will do? We will separate out, we will remove this DNA fragments from this gel or from this agarose bed. Okay, so what is required? The separated DNA cut from this agarose gel. So what we will do? We will take out these DNA fragments from here, as it is visible now. So the method by which we can remove out this DNA fragments from the agarose gel, it is known as elution. Okay, this question is important. This question is important. What is elution? The removal of the DNA fragments from the agarose gel, it is known as the elution. Right? Then, as we will get these DNA fragments, these are the purified DNA fragments. Okay, and now we are having number of copies. With the help of this method, we have amplified the number also. Okay, and then we can use this DNA fragments in the recombinant DNA technology. So this is the method what we have to do. First we will take the restriction enzyme, then the use of electrophoresis. Okay, now in the next part we will understand that how we are having different types of vectors. Okay, if I have to incorporate the gene of any plant, what type of vector I can use? If I have to incorporate gene into the animal cell, so we are having different types of vector. So we will discuss in the later session. Right? So that's all for today. Thank you.